and welcome to today's lesson on National 5 Biology, Unit 1, KA3, DNA and the production of proteins. Before we begin this lesson, let's do some starter questions. Pause the video here and try the questions and play again when you're ready for the answers. So where is DNA found in the cell? DNA is found in the nucleus. What's the function of DNA? Well, DNA codes for proteins, but you might not know this yet, and we're going to come back to that later in the lesson. You could also say at this stage that it contains genetic information, makes you you, although that's not a National 5 answer. For number 3, listing anything else you know about DNA, this was just to get you thinking about anything else you might have covered in science or biology before that was referring to DNA. We're not going to mark it just now, as there's so many possible answers that you could have put, but hopefully some of those will be covered in the lesson. So at the moment, we are still on Unit 1 Cell Biology, and we are on KA3 DNA and production of proteins. This is just a reminder to always check your Nat 5 mandatory knowledge or core spec document that's produced by the SQA, because this lists everything that you need to know for the tests or exams. This slide shows all the information that we're going to be learning about DNA today. So our learning intentions and success criteria today, we're going to try and learn about DNA, and I want you to be able to describe what DNA, chromosomes and genes are and how they relate to each other. I want you to be able to describe the structure of DNA, name the bases that make up the genetic code, and finally explain how mRNA and the ribosomes are involved in protein synthesis. Now, this diagram details how DNA is stored within the cells of our body, and as we go through the next few slides, you should be able to eventually come back to this and explain what this diagram shows. So our first big question is, where is DNA found in our cells? And as we know from the starter, along with Unit 1, that the nucleus controls most of the cell's activities, and that is where DNA is also stored within animal cells, plant cells, and fungal cells. Now, please remember, bacterial cells do not have a nucleus. So their DNA is stored as a bacterial chromosome and as plasmids, which is slightly different. Now, if you imagine the size of a cell in your body, remember we need a microscope to be able to see them. Now think about our diagram of an animal cell that we drew in KA1 and the nucleus inside of it. That nucleus is even smaller. Now DNA is really, really long, so it needs to be wrapped up tightly and organised within the nucleus so it doesn't get tangled or muddled up, especially when the cell is dividing, which we're going to come back to in a later topic. So to do this, the DNA is tightly coiled into structures that we call chromosomes, just like this one here on the right-hand side. Okay. Now, in the human body, our cells, each one has 46 chromosomes in its nucleus, and you can see them here. So slightly different sizes and shapes. Now, in the real cell, they wouldn't be neatly laid out like this. You'll also notice that these ones are long lines compared to this one, which is an X shape. Now, you don't need to understand this at the moment. We're going to come back to this when we're covering mitosis in a later topic. I just wanted to show you them both at the moment so that if they come up in future diagrams, you know that they're both chromosomes. So the nucleus contains chromosomes, which are made up of lots of coiled up DNA. Now, chromosomes are sections of the DNA that contained within the chromosomes can be split up into sections or bands, and these bands are called genes, just like this one here. So in this diagram of a particular chromosome and the DNA that makes it up, so this is the chromosome, this is the DNA. If we uncoiled it, you can see that this bit of DNA contains two genes. So this section is called gene 1, and this section of DNA is called gene 2. Now, you have to know the definition of a gene. It's a really common question in the exam. A gene is a section of DNA which codes for a protein. Proteins are essential in cells for them to work properly, and they're also essential in our bodies. They have jobs including the roles of hormones, enzymes, and antibodies. And again, we're going to come back and do a whole topic just on proteins and their jobs later. Now, each gene codes for a different protein that the cell needs, and a single chromosome contains thousands of different genes. And remember, I pointed out the nucleus of our cells contain 46 chromosomes, so just one of your cells contains 46 chromosomes, each made up of thousands of genes capable of producing thousands of different proteins. So that's a lot of information. So let's imagine this all together now. So we have our animal cell detailed here, but this could have been a plant cell or a fungal cell. It contains a nucleus, which contains DNA, which is coiled up to make chromosomes. Sections of the DNA, this bit here, codes for proteins, and those sections are genes. So if we're going to go from biggest to smallest here, you would have cells nucleus, chromosomes, genes, and then DNA. Now, it's really important you understand this diagram and this setup before we move on. You need to know the difference between a chromosome and a gene and DNA before we move on to the next section. So now we're going to focus in on DNA by itself, so this coiled molecule, in a second. So DNA stands for, so this coiled molecule here, DNA stands for deoxyribonucleic acid. Now, I should point out that you don't need to know what it stands for. You will not be asked this in an exam, but it's just kind of nice to know it. So here are some key facts about DNA. So DNA is passed on from parents to their offspring. So essentially this means you get half of your DNA in each of your cells from your dad and half from your mum. And again, we'll come back to this when we do mitosis and producing new cells later. Everybody's DNA is unique, so none of the codes are exactly the same as each other apart from identical twins, but we're not going to go into that just now. Now, this next point is really, really important. You'll notice that DNA is not a long straight line. There are two strands. There's like a red strand here and a blue strand here. This colour is just for help. It doesn't actually, not that colour in a real cell. Now, not a long straight single line. There are two strands, so we call it double-stranded, and it also forms what looks like a kind of twisted ladder shape. Therefore, we say the shape of the DNA is a double helix. So helix for the twisted shape, double because it's got two strands. Double helix is a phrase you really need to remember. Now, if we think of this as a twisted ladder, what's holding the ladder together? What makes up the rungs? Well, these parts in the middle that go across here, these are called bases, and they come in pairs and they hold the DNA molecule together. And we'll learn more about these bases in a second. Now, before we move on, let's try some quick questions to check our knowledge before we learn any more. So try these 4 questions, pause the video, and then play when you're ready for the answers. So number 1, where are chromosomes found in a cell? Chromosomes are found in the nucleus. What are chromosomes made of? They're made of DNA. What do genes code for? Genes, remember, a section of DNA that codes for a protein. And number 4, what phrase do we use to describe the shape of DNA? It's a double helix. So now we're going to move on and we're going to focus in on the rungs of that ladder and have a look at those in more detail. So the bases that hold the DNA molecule together. Now, looking at the diagram here, there are 4 bases. There's A, C, G, and T. Now, their full names are adenine, cytosine, guanine, and thymine. You must learn the full names of these bases off by heart. Although some questions will allow you just to use the letters, a lot of them now you do need to write the full word. So looking at the shapes here, how do you think the 4 bases appear together to make the rungs of our ladder? Now, hopefully you notice that the adenine and the thymine could fit together if they were across from each other, and so could the cytosine and the guanine, but the others couldn't fit. For example, if we tried to fit the adenine one with cytosine or guanine with thymine, that wouldn't work because they're not the right shape, like you can see in the diagram here. So the most important rule about DNA bases is what we call they're complementary. So they only pair with one other base. The rules are that thymine always pairs with adenine and guanine always pairs with cytosine, like you can see in the diagram here. So A is always across from T, C is always across from G. Now remember, these are the 2 strands of DNA joined. So if we flattened it out from its double helix shape, it would look something like this. So a selection of A's, T's, C's, and G's on one strand, opposite their complementary base partner on the other strand. So as I said, A is always across from T, C is always across from G. And then this twists up to make that DNA we saw in the other diagram. Now you can come up with your own way of remembering the base pairs, but here are some tips. So you could possibly remember that C and G are curved letters when you draw them, so they pair together, and A and T are more straight letters, so they pair together. Or you could remember using a mnemonic, so you could think at the golf club, so at being AT, so A and T go together, and golf club starting with GC, so G and C go together. Now you can make up your own one for that as long as you can remember that those are the ones that go together. Now, we're going to use our knowledge about the bases that we've just learned about to look at 2 past paper questions that were in previous National 5 exams. So let's try this first one. So here's a multiple choice question which asks about which one shows the correct DNA base pairing out of the following 4 diagrams. So A, B, C, or D, pause the video here and try and figure out the correct answer. So hopefully you've worked out that D is correct, and that's because it has the correct pairings of A with T, C with G, G with C, and T with A. All of the others have a mistake somewhere with the incorrect base paired up across from the other one. Now let's try another question. So forensic scientists can take small quantities of DNA and use a process to make large quantities. Each molecule is separated and used to make two complementary strands as shown below. So these are the complementary strands, and then we have new ones in the middle pairing up across from them. It asks you to give the full names of the bases labelled 1 and 2 in the diagram above. So you want to know what is the name of this base and what is the name of this base here. Pause the video and give this a try. So for this one, you need to make sure, first of all, that you're giving the full names and not just the letters. You get no marks for putting the letters here. So for number 1, this base is across from G, so we know this must be C. And the word for C is cytosine. So the answer for number 1 is cytosine. Number 2 is across from A, so this must be T, so that's thymine. So cytosine and thymine are your answers, 1 mark each. So now if we were to pretend that this is just part of your DNA from your cells, and this extends out both ways. So let's imagine this is a little part of the DNA, and this keeps going on and on, left and right, and it's in one of your cells. And it's really long and coiled up into a chromosome. If I read along the bases, A, G, G, C, etc., the code would look something like this. So the code contained within DNA, the genetic code, is just made up of lots of A's, T's, C's, and G's, just like this, but going on for much, much, much longer. Now, the order of these bases is a little bit like letters we use to make up sentences, like our alphabet, but for a cell. And they have to be in the correct order to make sense to the cell, just like our letters have to be in the correct order to make a sense of the words. Now, the reason they need to be in the correct order is that the order of these DNA bases, so these A's, T's, C's, and G's, determines the order of things called amino acids. Now, amino acids are molecules that join together to make those proteins I talked about earlier that are really important to the cells. Proteins are just long molecules made up of lots of different amino acids, and it depends which amino acids are joined together, which proteins are made. So it all depends, as I said, which order of these amino acids there are, which protein is made overall. Now remember I said that genes, those specific sections of DNA, code for proteins. Well, it's the sequence or order of bases in those bits of DNA within the genes that determine which order the amino acids go in the protein. Like, I'll show you what this looks like. So every 3 bases on DNA codes for a certain amino acids. So for example here, we have one set of bases, TAC, which codes for this round amino acid, whereas this other one, so GGT, might code for this triangular one. And finally, ATG codes for the square one. So each amino acid is coded for by its own special sequence of 3 bases, and it depends on the order of these bases which amino acid you get. This is simplifying a lot, but what this is the order of those A's, T's, C's, and G's within the DNA in the gene determines the sequence of amino acids that then make up the final protein, like this one here. Now we know how the DNA bases within the genes code for the final protein. We now need to learn how it's made. So we know that DNA contains the genes which codes for proteins. Where do we find DNA in a cell? We find it in the nucleus. Now we need to think about where the proteins are made in the cell. So remember the ribosomes we learned about in KA3 cell structure? Well, those ribosomes are the site of protein synthesis, and they're way out in the cytoplasm, away from the nucleus where the DNA is. Now, I like to think about the chromosomes as being like massive recipe books for proteins, and they're kept within the nucleus. Now, each chapter or section of the book or chromosome is a gene. So the gene contains the base sequence or instructions for making the protein. Now, the chromosomes in DNA is really precious, and the cell keeps them safe inside this nucleus. So how are the ribosomes, which are all the way out here, going to be able to get those instructions to be able to make the proteins? All the way out in the cytoplasm, how are they going to get those instructions which are held within the nucleus? Well, the answer is they would make a copy. So that copy is called messenger RNA or mRNA, and you can see it here. The cell can make many copies of the instructions for a particular protein by copying the DNA base sequence from that gene. And that copy, which isn't as precious, can then move out of the nucleus, which is this bit here, into the cytoplasm and go to the ribosomes, where they can then use those instructions to be able to make that particular protein. Now here's a little example of all that information put together. So let's imagine that the sequence of bases of the DNA is a gene that's contained on one of the chromosomes in our nucleus, so this bit here. So imagine this is a gene. Now it codes for a protein that can't leave the nucleus to get to the ribosomes where the protein's made. So mRNA is made, which is a complementary copy of that bit of DNA, of that genetic code. Now, this complementary copy of the genetic code, this mRNA, can move out of the nucleus where the DNA is kept, and it can go to the ribosome, where the ribosome then adds the amino acids in the correct order to make the protein based on what these sequence of bases said in the DNA. Now remember, the order or sequence of DNA bases changes the order or sequence of amino acids and therefore the protein. So this DNA sequence here codes for these amino acids, so a particular protein is made. So in this case, we're pretending this is insulin. Now if I change the DNA bases, the order of these, then the order of amino acids changes, which you can see here, and a different protein is made. And this time, this might be a different protein like the one, something like amylase. Now, it's the same structure, the same steps that we go through to get to this, but because the base sequence is different, then that means the amino acid sequence is different, which means it's a different protein. So let's go through the stages of proteins from DNA again, because I know this is quite difficult. So number one, DNA is found in the nucleus in the form of chromosomes. Number two, a specific sequence of the DNA is a gene to make a certain protein. This sequence of bases in the gene is copied so that it can travel out of the nucleus, and this copy is called mRNA, or messenger RNA. mRNA takes a complementary copy of the genetic code from the nucleus to the ribosome, and then at the ribosome, the sequence of bases on the mRNA determines the sequence of amino acids in the new protein molecule. Now, it is really, really essential that you can describe those stages, as it often comes up as an extended response question, so it's worth quite a few marks. Now, try and remember to think about the DNA and then the mRNA and then the protein, and that'll help you think through all of those stages. Now, some other common questions you'll get asked on this topic are these ones here. So a lot of the time they ask about the function of mRNA, and your answer would be mRNA is a molecule which carries a complementary copy of DNA from the nucleus to the ribosome. This would be worth 1 mark, and you need to have all of this there. You need to say it's a complementary copy of DNA, and you have to say where it goes from and to, so the nucleus to the ribosome. Another common question they ask you to describe why the sequences of bases on the DNA is important for the protein. And remember, the answer is because the sequence of DNA bases determines the sequence or order of amino acids in the final protein. So let's try out some other past paper questions on these. So for number one, the bases in a strand of DNA make up the codes for the production of proteins. The DNA for every individual person varies. Describe the way in which this code differs from person to person. So pause the video here and give this a try. So the answer for this one is the order or sequence of bases is different. So that's why the code is different from person to person, because the order of bases would be different in each person. So a tip is, if you're ever unsure about a question on DNA, go for an answer like that. Talk about the sequence or order of bases and you'll usually be right. Next question. Name the single-stranded molecule which carries a complementary copy of the code from the DNA in the nucleus to the ribosome for protein synthesis. Pause the video here, give that one a try. So the answer to this one is just nice and short. They're just asking you to name it. So mRNA or messenger RNA. Now we're going to try and do some true or false questions. Again, these are really common in the exam. For each one, you just write down true or false. If it's false, you do not need to write out the whole sentence again. You just write the one word that you would replace the underlined word with to make the sentence true. So pause the video and try these and press play when you're ready to hear the answers. So number one is DNA is single-stranded. That's not true, that's false. And the answer would be double. So DNA is double-stranded. Number two should also have been false. So DNA is composed of 3 bases is wrong. It's composed of 4 bases. Number 3, the complementary strand for ATT-CGG is TAA-GCC. That's true, because each of these is the opposite. Proteins are made in the mitochondria is false, it should be ribosomes. And finally, proteins are assembled from amino acids is true. Now I'm going to finally use this diagram to summarise everything we've talked about in this lesson. So follow along as we go through. This is a really good diagram to help you memorise this topic. So if we start with chromosomes up in the top right hand corner, chromosomes are made up of tightly coiled DNA and are found within the nucleus. The DNA is a double helix structure because it's got 2 strands and it's in a twisted shape, and it's held together by complementary bases. Remember the bases adenine always pairs with thymine and cytosine always pairs with guanine. Sections of DNA which code for proteins are called genes. Chromosomes are made up of thousands of genes, and each gene codes for a different protein. This can be explained because the order of bases on the DNA within the gene basically determines the sequence of order of amino acids that are attached together to make the protein. So finally, we're going to try some quick questions on DNA. So pause the video here and answer the following questions. Number 1, during protein synthesis, what molecule is a copy of the DNA code? Answer for this one's mRNA, or messenger RNA. Where is mRNA produced in the cell? Be really careful, this is quite a trick one. So mRNA is produced in the nucleus, so nucleus is the answer, and that's because it's a copy of DNA. And DNA is found in the nucleus and can't leave, so the copy has to be made there, and then the mRNA travels out to the ribosomes. So nucleus for number 2. What organelle is the site of protein synthesis? It's the ribosomes. So there we go. So I hope this has helped you to learn about DNA and how proteins are produced in a cell, and that you can now basically check against these success criteria and make sure you know them all. Thank you so much for listening.